Science Uncut, presented by the Volkswagen Foundation.
I'm an HIV person, and when this gain-of-function story started to come to the fore, so this is in the fall of 2011, I was looking at it and it didn't jive. The claims or the benefits of this work didn't jive with my appreciation of HIV virology and HIV evolution. I've essentially concentrated on the molecular genetics and how the virus evolves and so on and so forth. And so I paid much greater attention, and I'm here now because I'm wondering about whether the science can actually deliver on some of the claims. But, you know, science is a tough topic, and we go down axes and they stop, but this is perfectly normal. This is my sort of subliminal slide because it's to remind ourselves where we're coming from. You know, we've been spending more than 100 years beating up microbes. So basically, if we make microbes more virulent, more transmissible, whatever, I don't have an intrinsic moral position. I just say we need some really good data if we go down that path. And these people and apologies to the ladies because there's an obvious gender bias, but this is a historical slide, so I hope you'll forgive me. And it's also on there because the gentleman all the gentlemen either isolated microbes or isolated microbes and made vaccines. And the gentleman, Stanley Plotkin, is smiling because he's the only one alive. So I'll come to Stanley. From the previous speakers, they gave you this very clear description of their experiments and the background of flu, and this means I can go much faster. So if we make a matrix of all the... Is there a pointer? Doesn't matter. A matrix of all the hemagglutinins, because the two major proteins on the outside of an influenza virus are H and N, hemagglutinin and neuraminidase, and there's 16 hemagglutinins and 9, well, there are some in bats, but this is in the aquatic bird reservoir. And in yellow, those are genetically confirmed isolates. This is no serology. We're 100% sure of all this. And the yellow in ducks, the orange in shorebirds. And so that's 127 different types of, generally speaking, HN combinations. Now that's the major reservoir. The anthropocentric part of the slide is on the top in black. Those are the pandemic strains. That's the data for humans. H1N1, or strains of that large type, H2N2, H3N2. That's 100 years of data, 5 pandemics, okay, in that period. And the rest is unknown territory. And of course, now with surveillance, and as Yoshi mentioned, surveillance is getting better all the time. And people are picking up these sporadic symptomatic infections. The asymptomatic, as Peter has mentioned, but we'll leave that to the side. But these are the symptomatic ones. And those shown in red, and you see H5N1, more than 700 cases, H7N9. And our surveillance is still not perfect, but it's pretty good, so that we can pick up one case of H6N1 in Taiwan. So these come over. And these are, we call them in virology, they're spillovers or they're dead-end infections. That is, they come over and they don't get transmitted, and they die if the individual dies, or they die if the individual recovers. You know of a very clear case of dead-end infections, rabies. Going back to Pasteur, there are 55,000 dead-end infections in the world, and if it's untreated, they all die. This happens. This is part of the microbiological world we live in. It's not very palatable, but it's fact. And viruses are spilling over, and with our modern technologies, we see all sorts of strange viruses that come in. So occasionally they're asymptomatic, but frequently they're asymptomatic, and often they are, and occasionally they're symptomatic. But it's not just that, the myriad of viruses. On this slide on the left, you have a genetic tree of H5, just the H5 hemagglutinin sequences, and you see this genetic heterogeneity. The arrows indicates the viruses that Ron, Yoshi, and Madam Chen used in their gain-of-function studies. we have a myriad of variants for each N, and then there are swine flu viruses. It can be important, and pandemic 2009 came from swine flu. And of course, just to make you feel happy, there's H3N8 in dogs. So the next time your dog licks you or whatever, think how your close proximity with those animals, but it hasn't come over despite this very close contact. So we've seen the experiment, and this is this. This is Ron's slide, the index infected animal on the left, the airflow, and will the receiver animal get infected? And this is the basic of it. Now this, Ron has mentioned that you passed this several times in some. It can be one passage for some viruses, but in his case, and there are more. And this is the equivalent to a selective screen. And this is where the scientist plays an important role. With even bacterial selective screens on a little plate, you select for what the screen is designed to select for. So if you then take in the receiver ferret, you take an animal with mild symptoms, and then you repeat this, you'll end up with a highly transmissible virus with mild pathogenic potential. If, of course, you took an animal with severe respiratory distress, and then you constantly selected for animals with severe respiratory distress, then you would have a highly transmissible, highly pathogenic virus. And the observer actually, the experimenter, chooses a little bit what the outcome he wants for individual reasons. But there is, we would say, observer bias. Now, I'll give you an example to help the non-scientists in the audience understand this. Here's something. Breeding, dog breeding, it's a selective screen. We breed dogs for various traits. This has been done over 10,000 years. The starting point is the wolf. And this is a dachshund. This is for the German part of the audience, and the rest is for the British, the English corgi, the bulldog, and whatever. But the English have this fascination for dogs. So we can do this in less than 10,000 years of evolution. The question is, which of those trajectories will nature take? Because when doing those experiments, we're trying to anticipate what nature will do. And so will nature follow any of these trajectories that will give rise to a dachshund or a corgi? Well, facetiously, I will tell you that the answer to that question is on the slide. Just look at the wolf's mouth. It's going to eat this dachshund, right? So there's no way nature's going to follow that trajectory. But of course, in virology, it's a much harder call. But this illustrates the fact that when you go down rapid passage and extensive passage with the strong hand of the experimenter, you go down trajectories, and the question is, will nature go down that path? This is an example, but to get the message across. But we have to get into the details of this. Now, I think there's ambiguity, and I can't go through all of this. The question of the observer bias. Will avian viruses evolve in a comparable way? This is a futuristic comment. There's a probability-based comment, and it's the future. And I don't want to quote Yogi Berra, but getting into the future is not always that easy. One interesting thing about evolution is this contingency. In a lay language, it's chance. Chance has always played a very strong role in evolution. There are multiple studies to show this. And of course, when you have the strong hand of the experimenter, the chance is not really in there. One point about the viruses, they were isolates made in 2001, 2004, and 2005. And we're in 2014. Would those trajectories, I understand the experimental setting, but will they be taken by, say, a 2013 isolate? There's considerable sequence variation between those viruses and those that are actually out in the field. Can we transpose knowledge from those strains to, say, contemporary viruses? And as you know, in evolution, you have lineages that go extinct and new ones that comes up. And it's the same thing happens in virology. Lineages become extinct and the new ones take their place. Can we transpose? The data on this is, we don't have much data, and as in science, we always want more. But it seems that in some of the mutations from Ron's lab, you put them into an Egyptian contemporary, or more, I think it's 2010, you don't necessarily get the same phenotype. So then we have to say, well, what we call the backbone in genetic terms, this is influencing the outcome. This is known in many situations. It occurs for human proteins. It's called epistasis. It's a known complication of our lives. But there is the question of the transposability. The ferret has its limits model. We heard from our speakers today, I come from HIV. We have a macaque model with a macaque virus, and it's very much more close, of course, the macaque to humans, but we are worrying about, does the macaque model faithfully represent and reflect what's happened in humans with HIV? And Ron is quite right, the best model for man is man. There's no doubt about it. And so the questions of models, we're always asking ourselves this. Of course, we will never be able to do an inoculation of a human being because it's unethical. So in some ways, in a Popperian sense of the word, the experiment is unfalsifiable. This is typical of animal model results. Can we transpose? Can we disprove our experiments? Well, we can predict. We think it can, but ultimately they're unfalsifiable. So we're left with this question of how, to what degree, the data derived from this these experiments will help us interpret what's going on in nature. And, you know, let's face it, there are enough microbes out there. We're lacking 34 vaccines to major pathogens. We need more vaccines. We've got a lot of work to do. So, you know, yes, we've got to take nature head on, but can we learn from these? And I have a few reservations on that. There was a recent paper out in Journal of Virology this year on an ostrich H7N1. This came from Dan Perez' lab, and he first passed it mechanically between ferrets, and then finished the study with a single transmissible airborne transmission experiment. The virus, which was highly pathogenic by infecting the animal through the mechanical route, just simply blowing it up the nose, it killed roughly 60% of animals. That pathogenicity, at least in the ferret model, was not lost upon acquisition of airborne transmission. So there was this, how could I say it, not actually hand-waving idea, but the rough idea that perhaps with the acquisition of transmission, you would lose pathogenesis. This paper shows that this is not the case. It could be, no problem, a different virus, a different strain, but it's not always the case. This is a highly pathogenic virus that kills 3 animals out of it. But the thing is that there's actually never been one confirmed case of H7N1 in humans. You see, here's the matrix again. And you see it's yellow for H7N1. There's not been one recorded spontaneous case. Recorded, that is lab confirmed. There might have been, probably, but we don't know. So therefore we do not have the data to say this is of major public health concern. It's not even one case. So you could say it's a surrogate marker for the H7N7, H7N9, would it not be better to do the experiment with those viruses, one of those viruses? So I don't actually understand the raison d'etre, apart from saying it can happen. But I don't see, yes, maybe when, we do not know. The benefits, as in the early stages, and Ron is quite right, you know, science is complicated, and we always find that it's more complicated as we proceed. But in the early days, it was said that this work would help us with pandemic preparedness, and say predicting the evolution of the virus or predicting the next pandemic strain, which would help us make stockpile vaccines and drugs and interpret mutations in the field. If we look at some of this, well, again, science is tough, but the expectation, I think, and it's a probability-based expectation, but from the flu community, was that the next pandemic would probably be an avian virus in Southeast Asia, and it turned out to be a swine virus from northwest Mexico. That just tells you how hard some of our work is. And no one predicted the emergence of H7N9 or H10N8 spillovers. H7N9 was totally off the radar, but now it's on, very much so. And this is a recent paper in Russell et al., which is a large number of flu virologists. And the first line of the conclusion is, it is not possible, with present knowledge, of course, it's not possible to predict when the next pandemic will be, which means if we can't predict the strain, we're not in a position to make really substantive information about can we make a proactive preventive vaccine. And I won't go into this, but my colleague Adel Mahmoud, who will address this issue. About drugs, well, if a pandemic came this season from nowhere, drugs would be a very simple issue. Is the virus sensitive? If it is, we say, terrific, oof, aren't we lucky? But if it's resistant, by the time we've got candidate drugs into phase 1, it will be seasonal flu. We can't just make antiviral drugs that fast. We would love to, but we just can't do it. So it's a very binary decision, and if hopefully it'll be sensitive, but we just do not know. Interpreting mutations in the field, I think much of this could be done by just testing the viruses that are out there. This is concrete data, it's prosaic. If there's a virus in Egypt, then we can study it. If it's got a certain subset of mutations, we can study it. We can take it to pieces. And we don't have to worry whether that trajectory is something that nature might go down, could go down, perhaps will go down. We know it's concrete because it's out there. The range of risks. Mark Lipsich is going to address this in great detail, and I won't trespass on his time. But if we did ever, and to touch the question posed by Harvey, if we say that an infection, flu, took out my son, for example, I would say, inshallah, c'est la vie, that's life. But if, being a virologist, and I could do PCR and barcoded the virus and found it came from a lab, well, you know, I might just go and see my lawyer. And I would love to know what lawyers think about this. No one's addressed the question of liability claims. It's man-made. If I get, if a rock drops on me, so be it. But if someone knocks me down in a car, I think we're interested in third-party insurance. There's precedent for this. We don't know, no one's addressed this, no one's thought about it. These are risks. I may be inventing them, my apologies, but I don't think we've heard from people. We haven't heard from insurance companies. I say this because I come from a private institution. Pasteur is a registered charity. Yes, we have an endowment, but I don't think we would survive. And certainly, in talking to donors who come by, making viruses more dangerous, that's something that it's a very, very hard sell to them. And I would imagine this is true for philanthropic organizations. So I think there's a chance that we might be undermining research institutions in some ways. And therefore, we have to really think hard about this. And of course, the number one thing is public trust. Public allows us to do basic research. We're allowed to work and invest time in ABC, and other colleagues will deliver XYZ. That's the deal. Basic research underlies and goes through to FGH, STU, ending up with drugs in vaccines in the market. And this is part of the deal. And I think if we've got to really think about, is making viruses more dangerous part of this deal, because I find it a bit of a hard sell in a private institution where we have large number of donors. It's not something they easily understand. But that could just be educative. Is there any guidance out there? We're all looking for guidance. We're a heterogeneous bunch of people, and actually there is some guidance out there. There's this statement from the Inter-Academies panel on biosecurity. It's 2005. And the Inter-Academy panel is a larger, broad association of probably 76 or 78 research institutions, academic societies in the world. There's the Dutch Academy, there's the Leopoldina, the Royal Society, and the US National Academy of Sciences, but there are many more, of course. And it says scientists have an obligation to do no harm. Wait a minute, that's not medics, that's scientists, okay? Well, no one ever told me that. I came across this myself. It then goes on to say that, and recognize that individual good conscience does not justify ignoring the possible misuse of their scientific endeavor. Most scientists, I think, would say, once I've published my paper, I've done my job. And this is saying, you're putting information in the public domain, you can't just say that's not your problem, because you put it there. There is a dimension. No one taught me that either. Then it goes on to say scientists with responsibility for oversight of research or evaluation of projects. I think that's the funders and their reviewers, pretty clear. Or publications, therefore the journal editors, should promote adherence to these principles and act as role models in this regard. The italics are mine. I've spoken to some journal editors, and they didn't even know about this document. They discovered it, and it's talking about them. The people who do know it are Academy members, and I think perhaps there should have been more pedagogy. This is guidance. There are 5 elements. I'm just picking on 2. And we have to realize that we've got to get this information down to the PI and the postdoc. I stumbled across it myself. No one ever told me about it. I found it on the web. So we do need education. Education of the younger people, and education of people of my generation even. I think it constitutes a hiatus in microbiology because we've always been trying to attenuate viruses, beat them up, eradicate them, and now there's this chance of maybe we'll go the other way. I don't think this futuristic, looking into the future card is very strong, but I'm a data-driven, evidence-based person, and in science we discuss. We don't impose, we discuss. And look at the quality of discussions. I don't think, for the moment at least, it's going to deliver anything robust to a health minister. It's qualitative information we're learning. Predictive value, with more studies, yes, but for the time being it can't deliver. And I don't think anything is, should we be informing the public of a new risk? Is there an ethical dimension? By that I mean, we're making viruses that never existed before. Okay, yes, as Ron says, our biocontainment is colossal. Very, very good, and far better than when I was young. I would hate to tell you some of the stories when I was a young virologist. But as Mark will tell you, it's not as simple as that. And when you do some number crunching, it's not absolutely that reassuring. I'm actually worried myself by the inertia that I've seen before this issue. We've been debating or discussing this for 3 years, and no one has even dreamed of doing a risk-benefit analysis. Now it's the US government who's going to do it. No one. What were the other learned societies thinking of? Or the foundations, the philanthropic foundations, or the governments? The learned societies, I must confess that they have been remarkably quiet. The Dutch have moved. The German Ethikrat has a big and long report. We can discuss the merits. We'll hear about this. And then the US has moved recently. But that's about it. And yet we're dealing with microbes that, well, that worry some people. And I think this, because maybe this inertia and the lack of face-to-face or discussion or other people sort of, even learned societies, commissioning a risk-benefit report to sort of undermine a little bit public trust in science. We've got public trust, but I think we have to work to maintain it. It's like democracy. You have to work, you have to fight to maintain it. It's not something that's given and we can assume it's taken. That's my personal opinion, and I'd love to hear from other people. And to help to try and understand, I think let's think of intent. This might help us sift between what is my last slide, what is and maybe problematic. People have said, well, this is just synthetic biology. No, I don't believe the synthetic biologists deliberately want to make a superorganism that could be dangerous to man. If they did, they would probably be horrified. They could, point. Here is to make something that is transmissible, a virus that is not, that is. And hence, if it resembles one of those 7 Dirk experiments, then I think we should flag it up. That's not to say we don't do it, but we discuss before writing the grant or doing the work. We don't do it afterwards. And if occasionally we go down this path, then I think it's very simple. We first do no harm, and second, we need very good reasons for doing so. I think they should be spelt out, and we should try and establish a consensus in the infectious disease community, because that's always been our strength in infectious diseases, that we've all been together fighting malaria and other things. So that's just a few suggestions as to why I'm worried or concerned, but I would love to discuss it further in a few minutes. Thank you very much.